Dear students, the protein folding process is essentially dictated by the amino acid sequence of the protein. And all the information that is required by the protein to fold into its native form exists within the amino acid sequence. So in this module, we're going to see how this entire folding process can continue and what are the different models that exist for this folding process to take place. As I just mentioned, that the information required for folding a protein into its native form is present within the amino acid sequence, right? And that the structure that comes out after the folding of the protein is thermodynamically very stable with the minimum amount of free energy. Considering these two things, I will introduce you the first model of protein folding. So this model is called the framework model. In the framework model of protein folding, what happens is that your amino acid sequence first forms secondary structures such as the alpha helices and beta sheets. We're going to study each one of them in detail later, but for now, you just assume that these different portions in the amino acid sequence, they form multiple secondary structures as shown here. So what happens is that the entire protein sequence, the amino acids, they form individual secondary structures. And these secondary structures, they are together in the form of a string here. So this is the framework model, which says that the amino acids first individually act to form secondary structures. So once the secondary structures are formed, the sub-organizations or these secondary structures, they come together with each other like that, and they make a complex structure. So in the first step, the amino acids, they make a secondary structure and are arranged. In the second step, the secondary structures, they are arranged to make a bigger suprastructure. So this is the framework model. Next, there is the nuclear condensation model. In the nuclear condensation model, the first step is that all the hydrophobic cores, they are collected and they form a nucleus. So therefore, it's called the nuclear condensation model because a nucleus gets formed first, followed by treatment of other residues. So the important thing to focus here is that the nucleus will essentially be composed of the hydrophobic amino acids, because the hydrophobic amino acids need to hide inside the core of the protein, and therefore if you form the nucleus, all of these nuclear amino acids will be hydrophobic. So once you form the hydrophobic core, the next step is to wrap around the nucleus, and the remaining amino acids that were left and that were not hydrophobic, they actually fold on top of the hydrophobic core. This leads to an expansion of the nucleus and increase in the protein 3D structure's size. So these two models are two approaches which predict how a protein can be folded. But we still do not have a definitive solution to how proteins are actually folded. So to conclude, several models exist for folding a protein given its amino acid sequence, and that the fundamental assumption and requirement is that the folding process remains spontaneous. It remains very quick, because in nature the proteins, they fold very quickly as well. And as I just mentioned, there is no definitive solution to this folding problem till date, and only these models exist which kind of estimate the process of folding.